Genetic engineering is an amazing discovery. It allows scientists to make very precise changes to an organism's DNA. This incredible technology has huge potential in many fields, including healthcare, agriculture, and environmental science. By controlling genes, scientists can bring out the best traits in organisms, fix genetic defects, and find new solutions to some of the world's biggest problems. Imagine a future without genetic diseases, where crops can adapt to climate change and environmentally friendly solutions come from genetically modified organisms. Genetic engineering offers a glimpse into this amazing future. From treating inherited diseases to producing drought-resistant crops, this technology can change our world. One of the most important discoveries in genetic engineering is the development of CRISPR-Cas9 technology. This incredible tool gives scientists the ability to edit genes with unbelievable precision, like using molecular scissors to cut and paste DNA. CRISPR-Cas9 has greatly sped up research in various fields, leading to groundbreaking discoveries and new medical treatments. However, traditional CRISPR technology has some limitations. It can sometimes cause unintended genetic changes, which raises questions about its safety and effectiveness. To overcome these limitations, scientists are constantly searching for more precise and effective gene editing tools. This search has led to the rise of a groundbreaking technology called prime editing, which is poised to revolutionize the field of genetic engineering. Prime editing is a new and more precise gene editing technology. It's based on the principles of CRISPR, but it offers greater accuracy and versatility. Researchers at the Broad Institute of MIT and Harvard discovered it. Prime editing has the potential to revolutionize gene editing, opening up new possibilities for treating genetic diseases and other applications. While CRISPR cuts both strands of DNA, prime editing uses a different approach. It uses a reverse transcriptase enzyme and a modified Cas9 protein. This ingenious system works like a molecular word processor, directly writing changes to the targeted DNA sequence. The prime editing complex includes a guide RNA, which directs the complex to the specific DNA target, and a modified reverse transcriptase enzyme that carries the new genetic information. Once the complex reaches the target, the reverse transcriptase enzyme uses the guide RNA as a template to create new DNA, replacing the old genetic code. Prime editing's ability to directly write genetic information into the DNA molecule, along with its remarkable precision, makes it a game-changer in gene editing. It has the potential to correct more genetic variations than traditional CRISPR technology. The potential uses of prime editing are huge and wide-ranging, spanning many fields, including medicine, agriculture, and biotechnology. In medicine, prime editing holds the key to treating various genetic diseases, offering a beacon of hope for patients and their families. Imagine a future where cystic fibrosis, sickle cell anemia, and Huntington's disease are no longer life-altering illnesses. Prime editing has the potential to correct the genetic mutations responsible for these diseases, offering a chance at a healthy life. Furthermore, the precision of prime editing makes it an ideal tool for personalized medicine, enabling treatments tailored to each patient's genetic makeup. Beyond treating inherited diseases, prime editing could revolutionize cancer therapy. By precisely targeting and modifying cancer cells, this groundbreaking technology could pave the way for more effective and less harmful treatments. Prime editing's ability to manipulate the immune system holds promise for developing new immunotherapies that boost the body's natural defenses against cancer. In agriculture, prime editing can create crops that are more nutritious, resistant to pests and diseases, and adaptable to climate change. This can help increase food security, reduce reliance on pesticides, and build a more sustainable agricultural industry. Imagine fields of crops that can withstand drought, resist common plant viruses, and provide increased nutrition, all made possible through prime editing. Along with the amazing possibilities of prime editing come important ethical things to think about. Like any powerful technology, it's super important to understand its effects and make rules for how to use it. Open discussions, public input, and countries working together are key to making sure this new technology is used responsibly. One of the main ethical worries about prime editing is the chance of unwanted outcomes. Even though the technology is really precise, there's still a chance of off-target edits, which means changes happen where they shouldn't in our genes. These accidental changes could have unexpected results for a living thing or its offspring. Another ethical point to consider is germline editing. This is when we change genes in reproductive cells like sperm or eggs. These changes would be passed down to future generations. This brings up worries about unintended results for future populations and the possibility of changing the human gene pool. Using prime editing to improve physical traits, brain power, or other characteristics instead of treating diseases raises ethical questions. These are tough questions with no easy answers. We need to think carefully and have discussions as a society. Part 5. Comparing prime editing and CRISPR. Prime editing and CRISPR are both revolutionary gene editing technologies, but they have some key differences in how they work and what they can do. It's important to understand these differences to see the unique advantages and potential uses of each technology. CRISPR-Cas9, often called molecular scissors, cuts both strands of DNA at a specific spot, guided by a small RNA molecule. This double-strand break starts the cell's natural DNA repair process, which can be messy and sometimes adds or removes genetic material by mistake. Prime editing, on the other hand, works like a molecular word processor, directly writing genetic changes into the targeted DNA sequence. It uses a reverse transcriptase enzyme and a modified Cas9 protein to make precise genetic changes without relying on the cell's error-prone repair process. This key difference in how they work gives prime editing a big advantage in precision and versatility. Prime editing can make a wider range of genetic changes than traditional CRISPR, including single-base changes which are hard to do with CRISPR. Also, prime editing's ability to directly write genetic information lowers the risk of unwanted off-target edits, making it a safer option. Part 6. The future of prime editing: advancements and possibilities. The field of prime editing is rapidly evolving, with ongoing research and development efforts focused on further increasing its effectiveness, precision, and applicability. Scientists are constantly working to improve methods for delivering prime editing components into cells, optimizing enzyme activity, and expanding the range of genetic changes that can be made. One promising area of research is the development of new delivery systems for prime editing components. Currently, the most common method for delivering these components into cells is through viral vectors, which can have limitations in terms of packaging capacity and potential immune responses. Researchers are exploring alternative delivery methods such as nanoparticles and lipid-based carriers to overcome these limitations and increase the effectiveness of prime editing. Another active area of research is the development of more efficient and versatile reverse transcriptase enzymes for use in prime editing. By creating enzymes with higher fidelity, efficiency, and thermostability, scientists aim to further improve the precision and effectiveness of prime editing, broadening its potential applications. Furthermore, researchers are exploring the use of prime editing in conjunction with other emerging technologies, such as directed evolution and artificial intelligence, to accelerate the discovery and development of new gene editing tools and therapeutic applications. These combined approaches hold immense potential for pushing the boundaries of genetic engineering and unlocking new possibilities for treating diseases and improving human health. Part 7. The wide-reaching impact of genetic engineering, a glimpse into the future. With prime editing at the forefront, genetic engineering has the potential to change not only medicine and agriculture, but many parts of our lives. From environmental restoration to industrial biotechnology, the applications of this transformative technology are vast and far-reaching. In the field of environmental science, genetic engineering can be used to develop bioremediation strategies, cleaning up pollution and restoring damaged ecosystems using engineered microorganisms. Imagine microorganisms that can break down plastic waste, remove heavy metals from contaminated soil, or absorb carbon dioxide from the atmosphere, lessening the effects of climate change. Industrial biotechnology can greatly benefit from advances in genetic engineering. Engineered microorganisms can be used to produce biofuels, biomaterials, and other valuable products in a more sustainable and cost-effective way. Imagine engineered algae-powered factories producing biofuels, reducing our reliance on fossil fuels and decreasing greenhouse gas emissions. Furthermore, genetic engineering can revolutionize the field of synthetic biology, enabling the creation of new organisms with specific traits and functions. This could lead to the development of new biosensors, biocomputers, and other groundbreaking technologies, with applications in fields ranging from healthcare to manufacturing. Part 8. Conclusion. Embracing the potential of prime editing. Prime editing is a remarkable advancement in genetic engineering. It offers incredible precision, amazing flexibility, and huge potential for treating genetic diseases and more. Like any powerful technology, it's important to move forward carefully, think about the ethics, and set clear rules for how it's used. The future of prime editing is bright. Ongoing research and development are pushing the limits of this amazing technology. As scientists continue to refine and improve prime editing, we'll see exciting new uses in medicine, agriculture, environmental science, and other fields, changing our world in profound ways. It's crucial to have open discussions about the potential benefits and risks of prime editing for society, so its development and use are guided by ethical principles and responsible innovation. By embracing the potential of prime editing while also addressing ethical considerations, we can use the power of this transformative technology to improve human health, advance scientific knowledge, and create a brighter future for everyone.